Right. I'd like to say thanks to Linda in particular for the invitation to be here, and I'm really happy to be at the Canadian meeting. With so many great scientists here and so much work, it's a great chance to catch up, and it's also a privilege to get to speak after Dr. Sammons, as a lot of the work I did for my PhD was actually in his lab in St. Louis to look at the increase in copy number of EPSPS and Palmer Amaranth. But as Linda said, she asked me to do a couple of things today. First of all, to speculate and think outside the box a little bit. And she also asked me to think about things other than herbicide resistance. So I'm going to do my best today to talk about all kinds of other things that we could do with genomics and weed biology that don't have to do with herbicide resistance. And of course, there are a lot. And let's see, it should be this one. Yeah. Now, first of all, when we say, what do we mean going down the rabbit hole? It's sort of a metaphor for something that might be confusing or something that might lead to some dead ends. We don't know where it's going. Nothing makes any sense. Even just Googling for images of down the rabbit hole is a big rabbit hole, let me tell you. There are a lot of things out there where people have come up with images. But that can be a little bit what molecular weed science can be, because there are lots of abbreviations and different techniques and new platforms all the time. And if you're not up on the lingo, it can be a little bit hard to follow, let alone sometimes it may not be clear just what the objective is, what the questions are, and how those techniques are going to get there. And again, that's what I'd like to address today, is to really kind of take this journey together as far as what are different things that are happening right now, what else is happening in the general world of plant science and genomics, and how can we apply that to our questions for weed biology. I really like this photo. What you're looking at here is the glass slide is from an Illumina HiSeq sequencer, and it's the next generation sequencing that I'll be speaking about a lot today. And then, of course, Palmer Amaranth in the background. And I think this kind of gives a metaphor for how can this technology and these approaches potentially give us some new insights to view and look at weeds in ways that we never have before. But this also comes at a trade-off. It's expensive, and we know that there's still a lot of work that needs to be done, hands-on, applied work with growers. And could this work actually be distracting from that? Do we need to be sequencing the genome of every species? Do we need to be training lots of students to learn how to do molecular techniques and genomics? Perhaps is that the right application of our efforts? And I'd like to convince you today that there are a lot of reasons why. And I think this is probably one of the biggest ones. As you all know very well, weeds are some of the best species at actually growing in our cropping systems, a lot of times better than the crops we're trying to grow. People have been battling weeds as long as they've been farming, for over 10,000 years. Even recently, there's been a documentation at some archaeological sites in Israel of weeds in some of those early trash dumps from archaeological sites where people are farming. So if you think about that, weeds are perfectly adapted to cropping systems and ever-changing. Some weeds have been re-domesticated, such as weedy rice has been kind of a bit domesticated and then gone weedy again. So you have all these traits of ferality. Weeds grow quickly. They make lots of biomass. They make lots of really cool secondary compounds, different things that might have biological properties, or different kinds of carotenoids, things that we might be interested in for nutrition. And it's all kind of there, it's just locked into, ooh, I've got a little bit more microphone there. That's probably good. Think about all the secrets that are inside these weeds, and how can we unlock them? And can some of these then potentially be applied to our crops to help improve those crops? So can we shine the light into the weed genome, better understand those, and then turn it around against the weeds to help us grow more competitive crops and more nutritious? The example I'd like to use today will be familiar to a lot of you, and that's, of course, kochia. It's a weed that we're working on a lot in Colorado right now. And as you can see from the map, it's widely distributed in North America. Very cosmopolitan type of weed, growing in lots of different systems, both agronomic cropping systems, of course, lots of non-crop areas. It's one of the first weeds to be growing in construction areas or disturbed sites. So what is it about kochia that allows it to do so well across such a latitudinal gradient? Think of the climatic differences between southern Texas up into northern Alberta. That's a big range, and yet this species is being successful everywhere. Well, some of the weedy traits that we know kochia possesses in particular, it is a summer annual. It also has C4 photosynthesis, which is, can be a very efficient mechanism of photosynthesis in warm temperatures. Does very well with hot weather, in drought conditions, and also in saline soils. But it also is a bit unusual among C4 weeds in that it does so well in the cold as well. It can handle freezing temperatures. It grows a lot of biomass, and it's very adjustable to different conditions. So again, I would ask the question, could weeds, kochia in particular, be sources of genetic traits that can serve to improve crops? Think about the seed production that a plant like kochia can produce. And a lot of other weeds are similar. You're looking at seed production numbers of 50 to 100,000 from one plant. For kochia, we know that this is one of the biological weak points in that it is a very small seed, not a lot of energy, doesn't live very long in the soil. It can be killed by burying. It will tend to not have a lot of dormancy. So those are things we may or may not want in crops, but certainly we could perhaps gain some new insights into other methods of control for kochia by better understanding the seed biology. This photo is really great. This is one that Dr. Phil Westra took earlier this year because, as you know, you get a lot of frost in the spring, and kochia is typically germinating early in the spring, even in late February or in March in Colorado. And what you're looking at here is a typical morning. There's a frost on these leaves, and this is the type of frost that would be fatal to a lot of our crops had they been planted and emerged and exposed to these kind of temperatures. However, kochia can just wait and later in the day be growing and really show no ill effects from this kind of frosting temperature whatsoever. And this seems to be fairly unusual for a plant with C4 photosynthesis. You won't find a lot of examples of this. So what is going on? How are they able to tolerate these cold temperatures? And that's one trait that could be very useful. We also know that kochia has lots of genetic diversity. You can see here from the flowering structures that the stigmas on the flowers actually tend to emerge first and are receptive to pollen before the anthers will burst and shed pollen. So this really helps enforce a lot of outcrossing in kochia and maintaining genetic diversity. So we know we've got a rich source of alleles and genetic diversity to mine if we want to go down this road. So another image that Phil Westra and Carl Rivet at CSU have been working on, this is with a scanning electron microscope. Where you're looking at here is a pollen grain that's been treated so that you can actually get the close-up picture of it. You'll see a couple of interesting things going on. First of all, it looks like a very aerodynamic pollen, doesn't it? Because it has that golf ball shape. And some work going on here, looking at how far the pollen can move. Part of the explanation may be this aerodynamic shape. But also those small circles are where the pollen tubes can emerge from when one of these pollen grains actually germinates on the stigma. So you can understand a lot about the pollination and the reproductive biology of kochia from looking at things in this species. But that brings us to, okay, these are, sound like interesting ideas. How can we actually do that? And one of the game-changing technologies that has happened is the incredible decrease in the cost of sequencing, just obtaining the sequence of nucleotides, be it DNA or RNA, over time. You might have heard before of Moore's Law. This is a principle from technology and computer science, where the rate of technological increase is doubling, I think about every 18 months is the prediction of that. And typically, sequencing does follow this rate of Moore's Law. So this is from up until about 2007 with what was called Sanger sequencing. The cost was coming down, But notice that in 2007, to sequence the equivalent of the human genome, which is 3 gigabase, 3 billion base pairs, was $10 million. So obviously that's an international consortium type of effort, an incredible amount of work, lots of people involved. And that's just to get the sequence data. That's not actually to do anything with it, try to understand it, do any studies to prove what's going on, just to get the data, $10 million. But then in 2007, what's known as next generation sequencing technologies were first introduced. And notice the incredible decline in cost very rapidly, to where now, in 2015, it costs less than $10,000 to get 3 billion base pairs of sequence data. Again, what are you going to do with it? That's up to you. But you can get the data for an incredible reduction in the cost. And that just opens the doors for so many fields of biology, and weeds are no exception. The problem with a lot of these is now everyone thinks of a great acronym for the next technique they're going to introduce, and you have RAD-seq and GBS and GWAS and whole exome capture and all kinds of things. And don't worry, it's not going to be painful. We'll have a lot of fun with this. But RNA-seq, these are all different things that are being applied based on the sequencing technology. Again, to illustrate the reduction in cost, this are some numbers actually from probably 2 years ago now, but it still illustrates the point. On the top line, you have the capillary kind of traditional sequencing costing 10 cents per 1,000 base pairs, roughly, if you're doing a pretty high throughput system. And that's pretty accurate, but you're paying a lot. But just see the orders of magnitude decrease in cost with different platforms. And the two that I'll be speaking about most are Illumina and PacBio. What you need to know about Illumina, this is really where the term big data applies. Illumina is that glass slide that I showed in the first slide. One run on that platform takes about 2 weeks, give or take, or maybe down to about 10 days in some cases. And it can produce anywhere from about 1 billion to nearly 2 billion sequence reads on one of those chips. Each of those reads will be 100 base pairs. So imagine 1.5 billion times 100, and that's how many base pairs of data you're looking at from one slide on one instrument in one lab in a week. And these things are all over the world, and they're churning out data. So not only are we getting these insights, but we're also having to learn how to deal with the computer science aspect of this. How do you handle terabytes and terabytes of data? How do you archive that so you can go back and utilize it again? Or what do you do with it if you don't need it anymore? How can you capture the useful aspects of it? So you have the technological side and the data management side. There's another platform called PacBio. This is sometimes called third generation, because this is a platform that actually sequences single DNA strands. Illumina involves a little bit of PCR amplification to get more copies so you can sequence it. But in PacBio, you kind of sacrifice the huge number of reads for a much longer read. Instead of 100 base pairs, now the current PacBio chemistry is claiming up to 20,000 bases in a single read. So that's very helpful when you want to try to put together a genome for the first time. However, there is a trade-off in accuracy. It makes more errors because it's using a DNA polymerase to run this. It works very quickly, and that's why it can do such long reads. But there will be some errors. So sometimes you can do a combination of these two platforms if you're trying to put something together for the first time. Some groups have had success with that. In fact, recently the Caniza genome of horseweed has been put together into a draft using a combination of Illumina and PacBio sequencing. As I have said now several times, the advantages of these approaches are you get lots of data and it's cheap, or at least much more affordable. But there are definitely still some bottlenecks, particularly when you're thinking of your favorite weed species and trying to work on it for the first time to get some genomics or transcriptomic information. How do you take all of these pieces of the puzzle that come from the Illumina sequencer, they're just short reads, you have no idea how they fit together, and actually solve that puzzle and assemble it into the correct sequence? Well, that takes a lot of computational biology and bioinformatics. some big processors, lots of servers running overtime. Then once you get the sequence together, you also have the issue of figuring out what it is and what it does. Now we can draw a lot of information from model systems, particularly Arabidopsis, where people have already gone through different sequences and they've made predictions on what they think this protein does, and they've actually done experiments to verify the functions of those proteins. So a lot of the Arabidopsis genome is functionally annotated with some proof. But a lot of times when you do a de novo, a brand new assembly of a new species, you just align your sequence to that database and see if you get a match. If you get a pretty good match, then you think, okay, that's probably what it does. And that's called annotation. That's a big step forward, but you still have this kind of biological validation to really understand what's going on. And we know another thing about weeds is that they often have pretty large genomes. One of the main reasons the Canyza genome, as you can see here, has been finished first is that it's pretty small. It's almost as small as Arabidopsis. So that's pretty advantageous. Palmer amaranth is probably at least 4 times the size of Arabidopsis, but still relatively small as far as plants go, because you can see some of the bigger plant genomes are even bigger than human. Here you have blackgrass, Alipocurus, at over 4 billion base pairs in its genome. Let's not even talk about wild oats. I think that's around 16 gigabase or something. So that just becomes even more of a challenge to work with. Kochia, fortunately, is a bit in the middle. I don't have it on this plot, but it's at about 1 gigabase for the Kochia genome. So a little bit more workable. I also want to point out this graphic I'm going to have a few times. This refers to what's known as the central dogma of molecular biology. And this is the idea that the genome contains DNA, and those genes direct the transcription of RNA, which is the single-stranded molecule, fairly short-lived, and that RNA then is used to translate into protein. And then the proteins are generally what builds the things and does all the metabolism in the cell. When we're talking about molecular biology, we can measure any of these 3 things to look for variation. We can look for differences in the sequence of the DNA. If we're talking about RNA, we can look at how much there is of a certain RNA or the sequence of it. And for proteins, we can look at how much there is, how its sequence, how it's working, look at all the biochemistry. As Doug showed with ALS, you can measure all the biochemical parameters. So I'll try to kind of have this on the slide just to point out which part of this process I'm talking about. Because the genomes are so big, however, I showed you the cost of sequencing is coming down, but that assembly really is a big problem. I'll show you in a little bit our work so far to assemble the Kochia genome. And putting together the genome is really difficult because you have the gene space, but you have lots of other stuff as well on all the chromosomes. And if you're interested just in the RNA, in the sequence of those genes and how much there is, sometimes looking at the transcriptome is really helpful. So the transcriptome is all of the genes that are expressed as mRNA at a given developmental point in time. There are lots of different transcripts, but you can sequence these for a pretty affordable price, usually in the range of about $5,000 to $15,000. You could pretty much take your favorite weed species, sequence the RNA, and get a fairly reasonable transcriptome put together. And this is something that you could do in the matter of probably around 3 months, as long as you can get good RNA out. So it's much more achievable than a genome. Then you can resequence different RNA samples to either quantify the expression of all those transcripts or look at variation in their sequence. That'll cost you on the range of about $1,000 to $3,000 per sample to get the data, and then you need to have some computational biology ability in order to run all those alignments and quantify the reads, so that's the bioinformatics part. But certainly working with the transcriptome gives you a lot more return for your dollar than working with the genome, especially if you just have some initial questions about what's going on. There are definitely some successful examples of this in weed science. In particular, the USDA group involving a number of different scientists in North Dakota, they've done a lot of work on leafy spurge, euphorbia, and they're looking at things like vegetative bud dormancy and germination of seeds. They've also done a lot of work on Canada thistle to understand, again, the release from dormancy from the vegetative buds, how growth is initiated in the spring. So a lot of basic biology around these perennial species, they've had some success by looking at the transcriptome. So that certainly gives some hope. This is what the transcript quantification can look like if you were to actually take the data and visualize it. What you're looking at here is one of these transcripts that's been assembled out of a transcriptome. So it's just a linear nucleotide sequence. Then all of these little bars are those individual reads from Illumina that have been aligned to that transcript. Then you have two different sample types here. In sample type number 1, there's lots of reads that are aligning to that. So that tells you there's a lot more mRNA of that transcript to start with in that sample because you've sequenced it a lot. Whereas in a different sample type, there's just a little bit. And therefore, you could conclude, okay, for some reason in sample type 1, this transcript is being expressed a lot more. And again, this process just involves sampling the tissue, getting the RNA, and then sequencing it on Illumina. So again, it becomes a lot more approachable than it was even just a few years ago. This is what we found so far with the Kochia transcriptome. This is a paper published by one of Phil's graduate students earlier this year, Andrew Wiersma. And he did one of these experiments. So he took Kochia samples, collected the RNA, and assembled a transcriptome. And it turned out, again, fairly well that we captured a lot of transcripts, so about 34,000 transcripts in total, representing almost 37 megabase of nucleotide sequence. The average length of these contigs was a little over 1,000 base pairs, which is about what most genes are. And a lot of these were able to get an annotation. You've got 82% matching something in the UniProt database. So something that someone else before has said, yeah, we found this, and it seems to be this kind of protein. So this is encouraging. Now, this is a resource that can be used for these types of experiments. So if you're working on Kochia, you don't need to even do this initial step. You can use this transcriptome and align your reads to it. But it's the sort of thing, it's not static. This can always be improved. You can take another step and keep making it better. However, because this is the rabbit hole talk, I've got to be very honest that the transcriptome isn't always the answer, because what are you measuring? You're measuring the RNA. And variation in RNA doesn't explain everything that happens in biology. Some things are explained by that, but some things aren't. And we know the transcriptome is also very dynamic. It's constantly changing in response to circadian rhythms, so day-night fluctuations. All kinds of environmental cues will change the transcriptome. Obviously, it's developmentally regulated. The expression of genes in the seedling is not the same as it is in the flowers or in a seed. So you've really got to think about what kind of RNA do I want to sample at what time? What will that look like if you have some kind of treatment, a stress treatment? You may have various ranges of responses that are happening after that treatment, and how can you capture those? And the results can get confusing, and the answer may not even be there at all in the transcriptome, if in fact whatever you're interested in is not regulated by changes in the transcript. We know lots of things can happen to proteins that regulate their activity, and you'll never even see it in this transcriptome. But don't get depressed, because you can still get a lot of good information. This is an example. This is a study from Christophe Delis's lab in France, and they're looking at blackgrass, some responses, and just this gives you an idea of the complexity, even once you get this data and you get it all put together, how to begin to make sense of it, because you're looking at 20, 30, 40,000 different transcripts, all making different changes. And this is an example of a way to plot it. You can see what's going on. So they've got transcripts in lots of different categories here. For example, ATP binding or iron binding. And the different colors represent whether they're going up or down in the two different sample types or not being regulated. So you both need to kind of have a systems biology approach to understand regulation of all these transcripts in that context. And you also want to try to drill down to find out what individual transcripts are actually doing. So it's just, you tend to get a complex network of what's going on that can take a while to make some sense out of. This is another recent study looking at parasitism in dodder. So you know that dodder it will germinate and grow, but then once it finds a host, it forms a haustorium that will allow it to invade and parasitize that plant. So this lab was looking at how that process of parasitism happens. And again, so they're doing RNA-seq on these different sample types, and they've aligned it to their reference transcriptome. Basically, what you're looking at here, they were able to identify these different categories of types of genes that are either going up or down at different times. So this is before it sort of switches into parasitic mode, and this is when it's about to form the haustoria and actually in the haustoria. So they were definitely able to detect changes controlling cell division, cell regulation, lots of things going up and down. This is another interesting plot. This is a principal components analysis. So actually taking the data of all of these transcripts, putting it together, and trying to identify however many different axes are explaining the data. So again, that's where these different color schemes are coming from. They're able to find the loadings of these axes with principal components and then parse these out to their different biological sample types. So again, this is really great work. But also at the end of it, it's hard to say, here are the 3 or 4 genes that are definitely involved in parasitism in dodder. It's still not quite to that point. Definitely a big step forward in understanding. But you can see the complexity that comes from this kind of big data idea, trying to look at 40,000 transcripts at once and understand what they're all doing. In addition to the changes in transcript expression, you can also look at variation in the nucleotides themselves. And these are the kinds of changes that can result in amino acid changes in the protein that then may have some kind of effect on the function of that protein. What you're looking at here, again, is an alignment of some Illumina data to a reference transcript. And if you're to zoom in, you can see that at some of these positions, you're actually getting two different nucleotides. That's what the red and black dots are. And this represents variation at that spot in the sequence reads that you're getting. So it means this individual has probably 2 different alleles. That information becomes very powerful if you want to start trying to associate a certain genotype to a trait that you're interested in. This is what that calling can look like. For example, if at that position you see all C's and then a bunch of A's, you can call that a true polymorphism. Or you might think it's an error. You don't see enough variation that you're actually convinced that that's real. And then you also have, once you determine that you do have a polymorphism, you can assign that a haplotype. So you're saying here's allele 1 and allele 2 that I see either in this individual or across different samples. So this opens the next acronym I'd like to tell you about after RNA-seq, and that's genotype by sequencing, or GBS. Now this term is sort of an umbrella term. There are lots of different variations and flavors of GBS, but the idea is that you take the genome, so now we're working with DNA again, generally, and you will kind of subsample a part of it and look for these sequence polymorphisms. They're called SNPs, or a single nucleotide polymorphism. Again, just as a reminder, so at a given position, you might have an A, a T, a C, or a G, and you'll genotype individuals and use that information to tell you something. The nice thing is, because Illumina makes so many reads, you can put a lot of samples together and call these SNPs. And then in a non-model species, such as kochia or any weed you may be interested in, you can generate, in probably an optimistic scenario, more than 10,000 of these SNPs. Now, if you may be familiar with other types of DNA markers, you have to work really, really hard sometimes to get 5 or 6 or 10 working in a weedy species, such as microsatellites or RFLPs. So suddenly you go from a very limited set of information that's still telling you something to 10,000 markers and a much more affordable scale. I just wanted to give you a little bit of an idea of how GBS can work. And again, there are variations on this, but in this one, you're digesting with what's called a restriction enzyme. These are enzymes that cut when they see a certain sequence in the DNA. Then you're going to pull out a certain size of fragment that has that cutter on both ends. So this is why you can take that 3 gigabase genome or 4 gigabase genome, say, of Alopecuris, and reduce it down to really probably just a few thousand base pairs from each individual, because only a certain number of sites will meet this criteria, having the cutter on both ends and being a certain size. So you'll select those ones that are the right size, that have your cutter on both ends. Then you ligate these adapters, and what's really important is a barcode. So from each individual sample, say you have 100 individuals that you're working with, different populations, or maybe a segregating genetic population, every single one of those gets its own barcode, so you know that it came from that individual. Then you can mix all of these together and run them on one lane on the Illumina. Remember, I showed you that glass slide, it has the 8 channels. So just one of those channels will produce around 150 million reads. It's very normal now that people will put 100 samples together in one of those lanes and then eventually be able to call SNPs at, say, 10,000 positions. So you're looking at maybe $2,000 or $3,000 to generate 10,000 markers on 100 individuals. That opens up a lot of possibilities. And one of those is then what's called population genomics. So rather than population genetics, we're kind of looking at the whole genome now. So what you can do here is you use these SNPs to test whatever your hypothesis is. Maybe you are looking at introduction of an invasive species, and you want to understand, was it introduced once or was it introduced multiple times, and look at relatedness between populations and see what kind of tree it fits, whether it's consistent with a single introduction or multiple introductions. So those are when you're sampling across the entire genome, you really can get quite a fine layer of detail about this. This is another project that Anita Cooper is a graduate student in my lab. She's working on, and in this case, she's generated about 15,000 SNPs, and you can put this into neighbor joining trees, run principal components analysis. One of the things about this is that a reference genome is helpful, but you don't actually have to have one in order to do population relatedness with SNP markers, because you don't need to know how those SNP markers are related to each other on the chromosome, if they're linked to each other. If you do know that, if you do have the reference genome, it tells you a lot more. But this is nice because you could basically, again, take your favorite weed species, and several pipelines have been developed, but this one is called UNIQUE. It's from the group at Cornell and the USDA. So you basically just have a pipeline where you can assign these SNPs based on the barcoded Illumina data approach, and then start to build a tree on how different individuals are related to each other. You can go to another layer of detail, and I'll admit, this is definitely the rabbit hole part of the talk now, because there's another approach called GWAS, or Genome-Wide Association Study. This has been developed in crops, in humans, and it's a really powerful approach if you want to identify for sure the part of the genome that's influencing a trait. Remember, I showed you that the transcriptome sometimes is not that helpful. If the RNA is not actually, the regulation of RNA is not influencing your trait, you won't be able to learn much by studying the transcriptome. But the genome doesn't lie. If you find a part of the genome that is definitely genetically linked to your trait, there's something going on there that's going to tell you what's going on. So if you look at this, what I've got is just an example from a study. This is in Phaseolus vulgaris, or dry beans. And this is a species that until just a couple of years ago didn't have a reference genome. And yet, what they were able to do, they had 206 different accessions. These are representing a huge panel of genetic diversity, and they're looking at water uptake time. So it's an important factor for dry beans and cooking them is how long it takes them to absorb water. And it turns out there's some genetic variation for that, and it's heritable. So they wanted to understand what genetic traits were influencing water uptake in beans. So they took 206 accessions, and they had their reference genome. They did GBS, so to get lots of SNPs from across the genome, aligned it onto their reference genome, and did a statistical association with the phenotype they're looking at. And the result here, this is what's called a Manhattan plot. So this is showing all the SNPs on the genome. The bars are different levels of statistical probability. And you see these couple of dots up here that are really high. So that means that there are a couple of SNPs there that are just absolutely associated with their trait across all of these diverse accessions. And it's really, it's explaining about 25% of the variation in water uptake, whether it has an A or a G at this position. Now, it doesn't mean that there's a gene that has a SNP for an A to G that's what causes the changes in water uptake, but it means that part of the genome, right in its neighborhood, there's something that's causing it. So then they could go into their reference genome and see, okay, nearby, here are the 5 closest genes. Let's see what they do. Let's see what happens if we knock them out, or go on with those kind of questions. But you can really get to the answer. What I think is exciting about this is when we're thinking about our weedy traits, we can really do this. If we can put a genome together, we can just replace that phenotype of water uptake with whatever it is we may be interested in, seed shatter, cold tolerance, drought tolerance. If it's something you can measure as a quantitative trait, you can do this approach. And then in the worst possible outcome, you'll get some good SNP markers for that trait, and eventually you'll be able to determine the genetic basis of it. However, you do have to have the reference genome. That's certainly the drawback. And you also need a lot of different collections that vary for your phenotype. And I think this is a challenge for weeds. I often think about this in kochia. Okay, we want to study drought tolerance in kochia, right? It's so drought tolerant. But that means we actually need some kochia samples that are not drought tolerant. We need some variation for that. And that's hard to find. You could generate that, maybe do some kind of mutagenesis to perhaps knock out drought tolerance, or get a really big panel of genetic diversity that then you can find that variation, basically. And it's probably out there, but that's just a potential challenge. It also helps if you know something about how these different collections are related to each other. You don't have to know that, but it's definitely helpful. And then the goal will be to associate these SNPs with the trait that you're interested in. And again, this will require a lot of resources. You've got to have the reference genome, and everything kind of needs to be moving along in order to really make this work. Yet another approach that offers a lot of potential, and also I think really could be done in weeds, is a classic mapping approach. You may be familiar with QTL studies, quantitative trait locus. These have always been done with different types of DNA markers, such as microsatellites, but now you have the potential to have such a more fine-scale mapping of the genome in terms of the density of markers across a chromosome. And this, again, is just an example in plants. Apples, which are difficult to deal with genetically because they're trees and they live a long time. But what they did here, they had a mapping population that was an F1, and they did a bunch of those DNA samples and found almost 4,000 SNPs. They're looking at the color of the skin of the fruit. And again, they found this region on chromosome 9 that was very strongly associated with that change in skin color they were interested in. And it turns out there's a transcription factor there that they think is probably controlling that trait. You'll notice even at this particular region, they don't have the best density of their markers. That can happen at the telomere sometimes. There's just less, well, there's more recombination, but they're still able to find at least that this interesting gene was there. So what could we do for weeds? Well, we need the reference genome. But I think really this mapping and NGS type of approach can work. A lot of our weeds we can cross. We can make an F2 that would segregate. For example, if we had a kochia that was handled frost really well, and then a different kochia line that was very sensitive to frost, you cross these, you get an F2, and then you could potentially map it and find out what genes might be involved in that. What do you do once you have that SNP? There also are a lot of good advances in what's called SNP genotyping. So you might want to, once you've identified a SNP, you want to use it in a survey of a population, look at lots of samples, and you don't want to pay so much as you would pay for Illumina to sequence it lots of times. Well, then you've got lots of options. One is called CASP, and this stands for Complementary Allele Specific PCR. Complementary with a K. This was invented by a company in Germany, so they spelled it with a K. But so it's called CASP. What you're looking at is basically, it's an allele-specific PCR. You have one primer that matches allele 1, and it has a certain tag attached to it, and then a different primer that matches allele 2. Then you quantify how much of those two different alleles you're getting. If it's all allele 1, then you see all the samples here. If it's all allele 2, you get the samples here. If it's heterozygous, they show up in the middle. Now, I've labeled this as two species in a hybrid. This is one potential application. If you know there's a spot, such as in the ITS or in a certain gene, that tells those two species apart, you can do this, and it costs cents. I mean, this is so cheap. And there are plenty of other platforms as well. I'm not necessarily just endorsing this one, but this is really cheap, and you can do this on hundreds of samples in almost no time. This is a pretty popular platform in plant breeding. You can even have options to scale this up. There are ways that you can have genotyping chips that are custom built for the species you're interested in. So eventually, who knows, we could have a kochia chip that contains 10,000 SNPs, and we can tell you everything about that particular sample, what its resistance profile is, what its adaptation to different environmental conditions is, all based on the SNPs. So now I'd like to take you through what we've done as far as assembling the genome of kochia. A lot of people in our group at CSU are working on this, and we're also collaborating with Pat Tranel at University of Illinois. But in particular, Eric Patterson and Carl Reve are working on this. We've got a good resource in an inbred line that Phil Westra has been maintaining for a lot of years now. So this inbred line is herbicide susceptible, and the reason it's good is because an inbred line we expect to be mostly homozygous at nearly every locus in the genome. One problem for a new assembly is if you have lots of those SNPs at different places, the assembler doesn't know what to do with it. You basically can only assemble one strand of DNA. So we have this inbred line, and we thought, well, we can get a lot of Illumina data, 240 base pair paired in reads. Those are pretty long. We also did the scaffold approach to get bigger pieces. And we were hoping that this would work out well. You can see we have really high coverage of the genome, I think, 160x. But it didn't quite turn out that way. This is an assembler called AllPath, which is a good assembler. And you see here we got 682,000 contigs. The longest one was 11,000, and on average they were 400 bases. So we're talking about a 1 billion base pair genome, which we've split up into 400 base pair chunks. So that's not very helpful at all. And it tells you sometimes when you go down the rabbit hole, you might not like what you find. But maybe you can turn this fish into a tasty soup. That's what we're working on. So why did it fail? Why did that not work out very well? Well, this is one of the observations that we found that's really quite unique in terms of kochia that hasn't been seen in a lot of other genome assemblies. What you're looking at here, this is what's called a K-mer distribution. So the assembler takes those reads and it breaks them up into small words of around 25 letters, so 25 nucleotides, and it just starts building from those. But there's a quality control step that you do where you take those K-mers and you scan your sequence data and you see how often a certain K-mer shows up. And that's what this is a plot of, is so k-mers of this certain abundance, so k-mers that show up 50 times show up a lot. And typically what you'd see in a well-behaved genome, which kochia is not, you would typically see that k-mers show up a certain number of times, and you get a peak and it drops off. But here we've got a second peak, so there are k-mers that show up about 150 times or so, and then there are k-mers that again show up at a more abundance, and so there's some kind of enrichment there. Now what are these sequences? We think that this k-mer distribution is partly to explain why our assembly didn't work very well. There could be a couple of explanations for why this is happening. If this kochia line were in fact a polyploid, that could explain it, because there would be some sequences that show up twice as often or 3 times as often because the whole genome has been duplicated. It could be possible this line has been inbred. Kochia doesn't like to be inbred. It normally shows inbreeding depression. So perhaps that was just a big selection experiment for a polyploid kochia, that being the only genetic type that could withstand inbreeding. So that's one possibility. A second could be some kind of genome expansion. So maybe there are highly repetitive regions in these chromosomes that are duplicated 2 or 3 times, and that happens a lot. And that results in an overall larger genome size. The third explanation could be something that's happened in the past. So that means the whole genome has kind of tripled, and now it's in the process of kind of coalescing and reducing, but there's still a lot of redundancy. For the first hypothesis, true polyploidy, this turned out to definitely not be the case. We checked the chromosome number under the microscope in our inbred line, and it has the expected number of 18, matching a lot of other samples. There are reports of tetraploid kochia in the literature, but I'm not aware of any that have been reported in North America. So it seems that everything in North America described so far is diploid with a chromosome number of 18. And that's what we had. So that explanation is out. Second one, being a big change in genome size, also seems to not be the case. In order to see that k-mer distribution, we thought that we should probably need to see about a 50% variation in genome size from what we expected kochia to be. And we do see a little bit of variation. This is about a 10% variation in a bunch of different kochia collections. But this is somewhat within the range of error of flow cytometry, for one. And it's not atypical of what you might see in a lot of plant species. So it doesn't quite seem that just a variation in genome size is necessarily explaining what we're seeing. But then what if we start looking at other related species in terms of their k-mer distribution? What you're looking at here, it turns out that kochia, or in the genus Bassia, I can't quite see here, but I think this is it right here. Does that say Bassia or does that say Beta? So, or maybe, yeah, this is Bassia here, and then we've got, I think, Beta right here. This is sugar beet, so fairly closely related. And then we've got spinach over here, amaranthus over here, and Arabidopsis would be further away. So we wanted to look, these different genomes have been sequenced, and look at their k-mer distribution. And this is what we found. So if you do the same kind of k-mer analysis on Arabidopsis data, so well behaved, just a nice little peak, and then it drops off. The grain amaranth genome data have been released. And what you see here is that, again, you have a nice peak, and it's basically tailing off. But very interesting in sugar beet that we do see a similar pattern of this major peak, but then a couple of more minor peaks. And then, of course, again, here's our kochia data with the 3 peaks before a tail off. So it suggests there could be something going on maybe in this part of the Caryophyllales, that perhaps there has been an ancient, some kind of duplication within the genome, and we're still picking that up, but it's not entirely sure. But we can see there, some kind of sequence redundancy is happening. We know that one way genome expansion can happen is through copy number variation. And perhaps that's something that's just very common in kochia, is this variation in copy number. Right now, we're working on continuing our genome assembly with the PacBio platform I mentioned at the start. One of the challenges with PacBio is you need very, very high quality DNA much better than what you need for PCR, and even better really than what you need for Illumina, because it is sequencing those single strands. So we've done a lot of work to get that high quality and very long DNA from Kochia, and we're awaiting those results to see if we can do one of these hybrid assemblies, take those PacBio reads, correct their high error rate with our better Illumina data, and then hopefully come to an assembly that will work and then perhaps feed into some of these research goals as far as doing GWAS or doing a mapping study for a particular trait of interest. But it's definitely a work in progress. Something I'd like to emphasize is that gene copy number variation really is an important source of genetic diversity across biology. You know, first things that may come to mind when you think of sources of genetic diversity would be mutations in genes that change amino acids and change function of proteins, changes in regulation of genes, when they're turned on, when they're turned off, how they're spliced. Those are all very important. But it turns out a lot of medically important conditions can be explained by variations in the copy number of a gene in the human genome, and an increasing number of examples in plants where the number of copies of genes can really affect the trait. I'm not going to talk about the one example of copy number variation that I'm really familiar with, but there's this one that is also really, really cool. This is in soybeans, and these authors were looking at nematode resistance in soybeans, and they're able to find out that resistance was conferred when this particular locus that contains 3 genes, this is duplicated from 3 to 10 times in resistant soybean lines. So what you're looking at here, this is the genome of a nematode susceptible soybean, and this has this locus assembled as they expect in their reference genome. But this technique is fiberfish. It's a bit difficult to see here, but what you see is they have a red and a green probe on the gene, and they can actually pick up, or it's covering these 3 genes, and they can pick this up 10 times in a row. The expression of these 3 genes is increased in the resistant soybean lines, and it takes all 3 of them. Just overexpressing one doesn't work. So you can achieve quantitative resistance by duplicating a piece of DNA several times. And this is something that can happen just by a slippage of a chromosome, an error, and filling in of some extra copies. So I think just keep in mind that this is a potentially important source of genetic variation. Another thing I just want to throw out there, and I'm not aware of anyone working on the weed microbiome, but you may have heard in the news now, there's a lot of research going on into the microbiome in humans, in animals, in plants, in a lot of important crop plants. The microbiome is referring to all of the bacteria and fungi that are associated with eukaryotic organisms. So we've got an incredible number of bacteria in our guts, on our skin. Plants have them growing on their roots, as you can see here, sometimes even within the xylem. And it turns out that there can be some interchange, some flow of metabolic materials, flow of different signals. And there's some evidence that some of the microbes associated with plant roots can contribute to abiotic stress tolerance. So there's definitely ag biological research going on in that area. Can you actually treat with the right microbes and improve abiotic stress tolerance? But who knows? Are there certain microbes that are associated with certain weed species, roles in what those weeds are doing? Again, the study of the microbiome becomes possible because of next generation sequencing. Essentially what people do is they just take a swab of everything that's there, sequence it, and then they can identify the different species that are present. And you can see how the composition of those species might change between individuals or between treatments or in different locations. So again, it's all driven by this incredible reduction in sequencing cost. So I want to wrap up, and I think this is where I hope we can go, is from this kind of confusing state of the rabbit hole of so many different things, and really come to a clear direction ahead of how molecular weed science can help us better manage weeds and produce better crop harvests and yields. That's also really the goal, and and do all of this in a more sustainable manner. As we know, we've got to continue feeding more people on the planet and do so in a way that we can continue living here. So a lot of the things we'll be involved in this is the student training, improvements in bioinformatics, and working on genomes of these selected species. So I hope there's some kind of case here that investment in this type of research is important. I'd definitely like to thank, I've got a lot of great colleagues at Colorado State University, Dr. Westra and Scott Nissen, and Frank Dayan is joining our group next year. We've got a lot of great graduate students in our team, and that's certainly a big part of how we can move this research ahead. Finally, I'd like to make you all aware of a conference that we'll be hosting in Denver in May 2017. A lot of you I know were able to attend the Global Herbicide Resistance Challenge that was held in Perth, Western Australia in 2013. So this is a follow-up conference to that. We really want to continue to address this challenge of herbicide resistance, and I encourage you all to check out the website. We'll be announcing some great things about the conference coming up, and you can also fill out a pre-registration form there if you're interested. And finally, I'd just like to say thank you for your time. Thank you again for the invitation. It's really wonderful and a privilege to be here, and I'd definitely welcome any questions that you might have.